Hello and welcome back to Microbiology. Today we'll cover Chapter 12, which is antimicrobial treatments and really drugs to fight infection. So, whenever we talk about these drugs, we're going to actually address them as antimicrobial chemotherapies as the big kind of overarching umbrella. And so these are drugs that should basically destroy an infectious agent. Whether again it's a virus that's not alive or a bacterial cell that is, the idea is the big term is actually antimicrobial chemotherapy. And whenever we talk about this, again, it doesn't matter what our target is, one thing to understand from the beginning is that there will be no perfect drug. Every single drug out there has risk, has side effects, and precautions should be taken with any drug taken. And so understanding this going in really helps to frame why we do what we do as we go throughout. So whenever we talk about the ideal antimicrobial, there are lots of features that we would really want it to be capable of doing, like being toxic to only the microbe but not the host. This is pretty hard to do. We also want these things to be cidal versus statics. Again, cidals kill. We'd rather kill off the infection versus just stop it from getting worse. Again, next to impossible with viruses, right? We would want it to be soluble and be highly diluted in the body fluids. Again, we want it to remain potent long enough so that it can be used effectively, but also can eventually be broken down by the body. We don't want to see the development of resistance. We don't want to hinder host immunity. Again, there are lots of things here that are important in understanding why we choose what drugs we choose. So one of the bigger things I want to point out is there's going to be a lot of terms here. So make sure and be able to go through and look at the terms as we go throughout. So let's start off with chemotherapeutic drug, and it's any chemical used in treatment or relief or prophylaxis of a disease. And so the idea is anything used to basically eliminate an infectious agent. Prophylaxis, again, is the use of a drug to prevent imminent infection of a person at risk. The idea, again, this is imminent, so we can do this proactively to try to prevent that infection. Antimicrobial chemotherapy, again, was the big overarching term. This is, again, a drug to really prevent or control infection. So antimicrobials include antibiotics, which again are naturally being produced or produced through artificial means now that specifically try to destroy bacteria. So again, antibiotics is specifically a drug targeting bacteria. Again, the antimicrobial is covering everything else out there. Okay, so antimicrobials, everything, antibiotics, specifically bacteria. And as I alluded to with the antibiotics discussion, some of the drugs are natural, so we again call them natural antibiotics. Some of these things are going to be partially man-made, so we call them semi-synthetic. Naturally synthetic drugs are man-made drugs. Another big set of terms to know here is spectrum. Spectrum is really kind of what are we going to affect. So a narrow spectrum drug is only going to affect some specific microbes. So a small subset. A broad spectrum, on the other hand, is going to affect lots of different things. But some things to consider here. If we have recognized the problem, we should use as narrow spectrum as possible to kill or remove the problem. Using broad spectrum does have its time and place, but to use broad spectrums consistently creates additional problems. So, I mentioned that antibiotics did initially come from living sources, including fungi and bacteria. So really what these were antibiosis techniques that prevented other microorganisms like bacteria from growing. So antibiotics really, again, prevented bacteria from growing. And again, by doing this, the thing that made the antibiotic actually protected itself and its food supply. And so this was a natural mechanism that was used for a very long time. One of the things we should point out is the chemistry in antibiotics. Again, we will take the original drug and tweak it to make it chemically different and thus effective again. Due to things like antibiotic resistance, eventually bacteria become resistant to the drugs. But if we chemically alter the drug, we can now make it viable again. But we've taken a natural thing and man-made kind of tweaked it a little bit, and as a result we now call it a semi-synthetic drug. But full-on synthetic drugs are completely man-made. These are things that do not exist solely in nature. And so the idea here is synthetic drugs should have limited resistance, but ultimately, again, resistance is a problem that plagues us no matter what. So I want you to understand the bigger kind of overarching discussion that we're going to be having is resistance. With antimicrobial resistance, one of the things to understand is we need to kill typically just the problem. And that killing additional things can create resistance above and beyond. The other thing to consider is if we don't kill everything, what remains is actually the strongest and the worst for us. And so the idea here is we have to be very careful in deciding which drugs to use. So going through and following appropriate procedures or steps to determining treatment is absolutely necessary. So there are 3 major steps here. The first thing we have to do is identify the cause of the infection. Once we figure out the cause, it would make sense to test the drugs and see which one works best against said infection. Once we then have figured out a list of drugs that will be effective, we can then look at the medical condition of the patient and understand which drug would be appropriate for them. But as I said before, antibiotic resistance has been a growing problem, and it's because even in step 1, which is identifying the agent, we are even failing at a medical level to do that. Instead, we've been prescribing broad-spectrum drugs that should take care of the problem, but also other things that are not a problem. And so the idea is that's creating resistance in not just one category, but lots of categories. And that is truly, truly wrong. And in doing so, we create bigger problems for ourselves. So really, before any drug should be prescribed for long-term use, we should identify the cause of the problem. Again, before we use a drug, we need to know what we're trying to kill or remove. That means that we're going to have to typically take and pull samples. Again, it would be logical to pull the sample from where the issue is occurring. And so again, if it's a gastrointestinal issue, a stool sample would be great. If it's a respiratory problem, a stool sample isn't going to tell you crap. The idea here is we need to pull from the source of infection to understand what is the cause of the infection. Once we've again identified the cause, the next logical thing to do is to figure out which drug would work best. And so the Kirby-Bauer technique is a great example of how we can do that. With the Kirby-Bauer technique, this is a pretty standardized system that really allows us to screen different antibiotics against a pathogen. Since pathogens can be diverse, what we want to do is formulate a consistent system that we can use repeatedly so that we know, again, the results should be accurate. And so the idea here is Kirby-Bauer is a very, very methodical and precise technique. But that technique gives us true, honest answers in terms of which drugs are the best drugs to use. So how we do this is we need a special media type called Mueller-Henton, or MH. And Mueller-Henton is unique in that it's very porous, because what we're going to do is we're going to stick tiny antibiotic-soaked discs on the surface of that plate after we spread bacteria across it. And the idea is the antibiotics will soak into the plate and spread out, creating a zone of inhibition where we prevented bacterial growth. That zone can then be measured and compared for the standard to understand which drug is working best. And so we can see exactly this. Notice in the picture we have this lawn growth where we again tried to spread bacteria evenly across the whole surface. Then we applied the antibiotic discs to the surface of the plate. And then after we have allowed for growth, we can measure the zone of clearing from one edge to the other edge to accurately measure to see which drug works best. So notice, let's look at the picture down here, the artist rendering. We will have basically a zone of inhibition. And to measure that zone, we move from one edge to the other in millimeters. Again, this is the standardized feature of this test. Again, we're going to use millimeters to measure. And so what you do is you get your measurement, which is around, in this case, 7.5, so it's 27.5 millimeters. And so the idea is I can compare that to the known, which in this case is 22 millimeters or greater means it works. So we know this drug works very well against this particular bacteria. So enrofloxacin is a great drug for this particular infection. So here's an example of how we can again do this. So for each of the drugs put on, we would take a measurement of that zone of inhibition. So our example is for hypothetical Staphylococcus aureus here, okay? And so you notice for erythromycin, we had a measurement of around 15 millimeters. And so we compare that, is that greater than 18 or less than 13? Well, that's actually in between, so we evaluate this as being intermediate. But let's look at gentamicin next. Gentamicin had a measurement of 16 millimeters. Is that greater than 13 or less than 12? Well, it's greater than 13, so that means that that bacteria is susceptible or sensitive to the drug, which means that drug works against that bacteria. But what about this neomycin down here? Neomycin has a zone measurement of 12 millimeters. Is that greater than 17 or less than 12? And so the idea is it's equal to 12. And so that means that, again, we do not get accurate results to show that the drug works. So we say that the bacteria is resistant to the drug. One of the cool things that exists out there are these rapid tests. These rapid tests, the idea is they don't just tell you which drug, they do list it here, but it's at different concentrations. So you go down and you look at where that zone of clearing is. So not only did it tell me which drug to use, but at what concentration. Because otherwise we have to determine concentration through things like tube dilution tests so that we can understand the minimum inhibitory concentration, or MIC. The idea is, what is the lowest concentration that we can still use this drug and kill everything that we want to kill. And so the idea here, and just really pay attention to this left image, we take and put medias full of bacteria in contact with antibiotics, but we vary the concentration of the antibiotics. So the first tube where we don't see bacteria growing illustrates that minimum concentration to kill the bacteria. So at this point we've figured out, again, kind of the first two situations, what is the cause and what drug is going to work and at what concentration. But there are other considerations, like the patient. Where is the infection? Can that drug even make it there? Again, if the infection's in the brain, there are lots of things that can't pass the blood-brain barrier, and as a result, that drug may not work. Again, resistance is another thing to consider. Are there other things in that environment that would prevent the drug from actually working? So the idea is we must consider the patient in this as well. So the therapeutic index, I'm not going to stress a lot, but I do want to just mention what it is. It's the ratio of the dose of the drug that is toxic to humans to its minimum effective therapeutic dose. And so the idea here, we want to take and decrease risk as much as we possibly can, but still get the job done of killing the infection. And so the idea is there are times where we don't have much other choice. And so we're going to have to typically expose a patient to a drug that may put them at serious risk. Alright, but back to what I was talking about with this idea that we do have to account the patient. And again, the minimum inhibitory concentration, or even again that therapeutic index, these are things that do apply to this ultimate idea of the patient. But there are lots of variables in play. You know, even allergies are something we haven't talked about yet. It doesn't matter if penicillin was the best drug. If the patient's allergic to penicillin, penicillin may not be an honest answer for this situation. But there are also times, like therapeutic index would suggest, that there may be a situation where we have to put a person at some risk to fight the infection that may ultimately kill them. Other things to consider are underlying issues with things like liver and kidneys, things that again help and try to remove waste and detoxify. Ultimately, this stuff will become waste, and as a result, we need mechanisms to help remove them. So if the liver or kidneys aren't functioning, we probably can't use that drug. The other thing to consider is age. There are certain drugs that are used in very young people, but not very old people. There are certain things that would never be used in a pregnant woman due to the fact that it could physically hurt the offspring. The idea here is the patient still matters, and so we have to figure out the cause of the infection, what drug works best, but also what drug can the patient use. So ultimately, there are times whenever there's no perfect answer. There are lots and lots of situations, as cited in your text, that honestly, there will be honest problems that come out of this. There will be real things that are negative consequences. But it really boils down to we either treat the infection or the patient dies. And there are times where our treating the patient physically hurts the patient long term. So, let's talk about a new idea here, and that's the word selective toxicity. Okay, it's another term I want you to know. The word selective toxicity is basically trying to illustrate that we want to just be toxic against the bad thing. We don't want to be toxic to even our cells, our host tissues, right? And so the idea is selective toxicity is something to consider, but selective toxicity is really kind of hard. There are lots of things that we share in common with all cells, and as a result, there's risk in taking certain drugs. So there are some drugs with, again, excellent selective toxicity. Penicillins are great because penicillins specifically destroy peptidoglycan cell walls. And as a result, again, typically not a big risk to humans. But even with something like penicillin, there is natural risk. With any drug comes risk, and that's something that must always be weighed against the outcome of the patient. What risks are we willing to take to help the patient get better? Most drugs are in some way toxic, but some are far more toxic than others. Okay, so the idea here is if there's a shared feature, like say we're going to inhibit protein synthesis, well, now that drug has a real honest risk against a human cell because our cells make proteins typically very similarly to the way bacterial cells do. And so we must understand that there will be outcomes that are unfavorable if we take drugs that do pose a risk to even our cells. So really it turns into what is going to be our mechanism or mode of action. We talked about this in the previous chapter, this idea we need a target. What is the target? And so a good common initial target is the cell wall. If we prevent the cell wall, the bacterial cells typically die. And since we don't have cell walls as humans, again, not much risk to us. But whenever we start looking at things like inhibiting nucleic acids, like DNA and RNA, well, we have DNA and RNA, and we use them pretty similarly to the way microbes do. And so the idea here is there are risks, and those risks need to be considered. Same idea with inhibiting protein synthesis or even disruption of the cell membrane. And these are all things shared with microbes, so there's greater risk of basically toxic effects to our cells. Another example we'll discuss just briefly is inhibition of folic acid synthesis, which is a mechanism for really one of our first man-made drugs or synthetic antibiotics. So here's a picture illustrating everything that we were kind of talking about with a little more detail. I'm not going to ask you detail stuff. I'm not going to have you tell me, aminoglycosides can target specifically the 30S subunit. It's just not something I'm going to do, okay? We're going to talk about this at the higher level, that penicillins inhibit cell walls, or, your carbapenems inhibit cell walls. We're not doing, individual drugs. We're doing family level. So we've already kind of discussed this, but let's quickly remind you, a broad spectrum drug will work against lots of different things, while narrow spectrum should only work on a very small set of things. The idea is we should try to use as many narrow spectrums as we possibly can, because we typically just want to kill the cause of infection, not everything else that could be helping prevent infection. So here's this kind of comparison of the level of spectrum for different drugs. So notice things like isoniazides specifically work really good on mycobacteria, like tuberculosis causing pathogens, right? While something like tetracycline, on the other hand, tetracycline covers basically almost everything that's not TB. And so the idea here is we would ultimately want to use the more targeted things. If you had TB, we should probably give you drugs to treat TB, not a broad spectrum that would basically kill off everything but the problem. So whenever we do this, we're going to go through several of these different categories. One of the things I want to keep in the back of your head is we're going to do this look at these antibiotics at a family level. And so whenever we do this again, understand its family level. We're not going to talk about exactly how amoxicillin is different than penicillin, or how erythromycin works versus a macrolide. Okay, so just understand family. And as a family, penicillins target the cell wall. And really, penicillins have 3 different parts to them: your thioring, your beta-lactam ring, and then the variable side chain. So if we look at any penicillin derivative, you can note that we still have the beta-lactam, the thioring, and then the R group, which is again the variable side chain. And so what we actually change amongst the different variations of penicillins, like what's different between nafcillin and carbonicillin, is literally just this variable R group right here. The core must stay the same, that beta-lactam and the thioring. And honestly, that beta-lactam is probably the most important structure that we have. So what does penicillins do? Well, penicillins with that beta-lactam ring actually disrupt the cell wall, causing the cell to ultimately lyse. So again, do not look at all the subgroups. Understand that as a family, penicillins target the cell wall. Cephalosporins are another family of drug that's again going to do like your penicillins did and target the cell wall. In fact, they have that same beta-lactam ring that is going to be the kind of bigger feature here. And so the idea is cephalosporins target the cell wall. So you can see again that core to the cephalosporins, the beta-lactam, and an additional ring, and then the 2 variable regions or R groups. So again, do not memorize these at a subgroup level. Again, we do it as the big family. So cephalosporins as a family target cell walls. There are also again some other categories like carbapenems that target cell walls. Carbapenems are very powerful but also very dangerous. And so the idea is carbapenems are kind of that last line that we reserve for only those situations where we have no other choice. There's also these miscellaneous drugs, things like bacitracin, isoniazid, vancomycin. And really, I don't care that you know these overall. They're helpful in understanding some bigger ideas, but the individual drugs and what they do, probably not that helpful. I illustrated again that they could have some hints or details that make them helpful, like bacitracin, for instance, is one of the only antibiotics out there that doesn't require a doctor's prescription. This is something that comes as a main ingredient in Neosporin. Isoniazid is a very narrow spectrum that targets tuberculosis. Vancomycin is something that we switched to after we saw methicillin resistance develop. And so we went to fight MRSA, we started using vancomycin. But now we're starting to see things like Versa, which is resistance to vancomycin. So notice now we're shifting our target. So now we're going to talk about families of drugs that target protein synthesis. And one of the first ones here is aminoglycosides. Aminoglycosides target protein synthesis. So here's an example of streptomycin. Don't memorize these structures, they're not important. Again, just know streptomycin is part of, again, a family of drugs that targets the protein synthesis. A bigger idea I want to point out is protein synthesis is a broad topic. So understanding that if we disrupt anything along the process, the outcome, the protein, probably won't happen. So if we never allow the ribosome forming, then we don't get proteins. If we stop the messenger from moving through the ribosome, no proteins. If we actually prevent the translation where the messenger and the transfer RNA can't come together, no proteins. And so the idea here is different types of drugs can target different features all around protein synthesis there. All right, so again, your aminoglycosides target protein synthesis. Another example of drugs that target protein synthesis include tetracycline, or really cyclins in general. Cyclins target protein synthesis. One of the things I want to point out about tetracycline, though, is it's a very broad spectrum and causes lots of significant side effects, including tooth discoloration. If you've taken enough of this antibiotic for a long enough period of time, it physically stains the teeth, and they cannot be destained. You have to physically cover them up or live with the color change. Okay, but one of the bigger things with broad spectrum drugs is they don't just affect the actual problem, they affect all other microbes as well. And so these broad spectrums are going to disrupt the normal biota of the gut and make you more likely to have secondary infections or super infections like C. diff. Again, Clostridium difficile can survive tetracycline, but again, that now leads to a situation where it has all the resources and grows out of control, causing then the disease pseudomembranous colitis. So again, tetracyclines target protein synthesis. Glycocyclines, again a cyclin derivative, also targets protein synthesis. So glycocyclines, again, target protein synthesis. Erythromycin, also, so just mycins in general, target protein synthesis. These do have a unique feature in the macrolide ring, but again, I want you to know at a family level, erythromycins target protein synthesis. So you can see again myosin derivatives here, the macrolide ring, moving on. So again, macrolides or the myosins target protein synthesis. There's again some other miscellaneous drugs that target protein synthesis. Again, don't stress on these at all. We mentioned this briefly, but the targeting of folic acid is a very unique system found specifically in sulfa or sulfonamide drugs. Sulfa drugs are going to specifically disrupt folic acid synthesis. This mechanism did not come from bacteria or fungi. This was something straight up conceived by us as humans. And as a result, we thought this was going to be the thing that saved the world, that by disrupting folic acid synthesis, we could kill any bacteria that was causing issues, and that we wouldn't see any resistance to this. But just like with anything that we've seen, resistance will develop. In fact, the bigger idea I should point out here is the more we use antibiotics, the less effective they become. So it doesn't matter if this was a completely man-made drug, the more we use the drug, the less effective it became, because again, more and more microbes would be exposed that led eventually to resistance. Sulfa drugs, as we said before, are going to be focused on disrupting folic acid, typically folic acid synthesis. So in general, again, sulfa drugs, folic acid synthesis. So here's an illustration of different sulfonamides. And let's move on now to things that target DNA and RNA. With really the fluoroquinolones. Fluoroquinolones are rather potent and specifically disrupt DNA or RNA. These do have some unfortunate side effects, including seizures and brain disruptions that can be very problematic. And fluoroquinolones target DNA or RNA. There is rifamycin, that again is another DNA or RNA targeting drug, but again, don't stress too much on this. So the next target would be cell membranes, and really this is our polymyxins. And so what we see is polymyxins came from Bacillus polymixa. This is again a bacteria producing an antibiotic to fight other bacteria, which is unique. One of the things I want to point out though is polymyxins are quite bad for us in terms of the ability to pass them. They're very toxic to the kidneys, and so we take great concern in using them. So again, polymyxins target the cell membrane. So now that we've covered again the different families of drugs and what they do, again I want you to know family level. Let's talk about kind of how they affect the bigger overall idea of things like biofilms. Whenever we look at bacteria independently, a drug may actually work quite well. But whenever they group together in biofilms, drugs become basically useless. And that's an important thing to understand, that biofilms offer protection to the bacteria that live there. One of the things that we have discovered is that we can prevent biofilms by disrupting quorum sensing pathways, but we could also increase the likelihood of antibiotics working if we pre-treat the biofilm, giving us now the opportunity to allow the antibiotics to get in. So the next thing we're going to do is move on to antifungals. And just like before, I want you to know kind of just a general idea. And so really, we're going to even be more general, and that I just want you to understand that fungi are eukaryotic, just like you and I. But I want you to understand that fungi also have typically cell walls, and so we can often use the cell walls as our target for our antifungals. Whenever we see the antifungals, they come in a variety of different things, like azoles. Azoles is very commonly used. Whenever we start talking about the antiprotozoan and anti-helminth drugs, again, the available drug list gets much, much smaller. And the more similar the pathogen is to our own cells, the more difficult it is to treat them without actually hurting our own. And so the idea here is the list for drugs actually gets often quite smaller, but then also becomes much more risky in terms of side effects. So antivirals are the next topic that we're going to cover. With antivirals, they typically take and block either the ability to get into the host, or they block the ability to transcribe and then translate viral information, or even prevent the maturation of the viral particle. Different antivirals will do different things. I don't care too much that you know each of the different examples here of the antivirals. What I want you to really understand is antivirals are going to be more of what we call a static versus a side. Again, side means to kill, and we can't kill viruses, but we can stop them from reproducing, which is again a static technique. So really, most antiviral drugs are going to be things that don't necessarily fix the problem, it just stops the problem from getting worse. And after enough time, again, they can typically clear the thing on our own. In the last slide, I mentioned that we can usually hold the infection off until we can clear it on our own, but there are some things that we may not ever be able to clear, like HIV. And so the idea is we need to prevent HIV and these other things from getting worse over time. And so routine use of static drugs in this case would be very helpful. And so again, viruses, if we can clear them, the body's the easiest and best way, but there are some things that we may need medication for to get things under control. So the next thing that we have to talk about is actually an incredibly important topic, and that is drug resistance. Drug resistance is going to be the adaptive response of a microorganism as it's exposed to potentially drugs. Okay, now I'm not really talking about individual cells per se, but the species overall. So let's kind of point out the general trend here. So whenever we talked about things like mutational error and this idea that evolution is driven basically by advantage that was created by the environment. So let's talk about drug resistance for a minute. With drug resistance, we're creating a hostile environment for the bacteria. And so whenever we use the antibiotics and we don't kill everything, what's really left behind? What's left behind is the thing that is resistant to the drug. So what's now going to be more common as time goes on? It's going to be the thing that was resistant. And as a result, what's going to happen to the population dynamic then? What's going to be more common the more we use the drugs? Well, it's going to be the drug-resistant microbe. So you see literally how applying this artificial pressure is creating the problem we currently have. And so the issue is really bigger than what people realize. With antibiotics specifically, the more we use the drugs, the less effective they become. So the answer may not actually be antibiotics, but rather some other form of treatment. Because if we don't kill everything with the drug, what remains is the strongest and most resistant. So let's talk about intrinsic versus acquired resistant. Intrinsic would be the natural resistance that happens whenever, again, a bacteria is producing an antibiotic itself. So again, there are some bacteria out there like Bacillus polymixa that can produce an antibiotic, polymyxins, to fight other bacteria. But if that bacillus wasn't inherently protected against the polymyxins it produced, it would be killing itself, which is really stupid. So there's always examples of intrinsic resistance. But what we're more concerned with is the acquired resistance, which is the narrative that I just painted a minute ago. This idea that the more we use the drug, the more we force the microbes to acquire resistance. And through things like horizontal gene transfer, the acquisition of resistance is not really that hard. The other thing to note is that resistance has to start somewhere, and that somewhere is spontaneous mutation. Just like everything else that we've discussed with evolution, mutation is the driving force for resistance. It takes one cell developing a mutation that offers an advantage against the drug. But the more we expose the microbes to that drug, the more we're pushing for that one resistant thing to become the normal. Or, in the example of genetics, the wild type. So literally the use of these drugs are pushing more and more resistance. And at a large-scale problem over enough time, we can see this become a huge issue that we just cannot overcome. So I illustrated before the possibility of resistance factors being passed through horizontal gene transfer. So things like plasmids can be passed through conjugation, transformation, or even transduction. Transposable elements have also been noted as means of moving drug resistance amongst sequences of genetic material, even to plasmids that then can be moved to other cells. The reality is resistance will happen, and the more we try to fight it, the more we realize that we're not effective at it. And so understanding that resistance is inevitable, and trying to figure out ways to overcome resistance may honestly be futile. Maybe the question really becomes, are the drugs the best option? Maybe we need to look at other options. Maybe we should use viruses to infect and kill bacteria. Or maybe we should use methods to promote immune response versus drugs that may actually hurt immune response. So as we've been, again, alluding here, these genes can be passed amongst organisms, but believe it or not, even amongst species. And so this can be passed at an alarming rate, where you can have one bacteria that's resistant that can pass that resistance on to another bacteria that's very different from it, creating a situation now where we have widespread resistance. And it doesn't just have to happen in the body. This can even happen in the natural environment. So we've talked about the bigger idea of resistance, but let's get down to some of the details of how this can actually happen. It can be as simple as a bacteria can produce an enzyme that breaks the drug. A good example of this is beta-lactamase. Beta-lactamase is an enzyme produced by some bacteria that simply breaks the beta-lactam ring of penicillins. And as a result, penicillins become completely and utterly useless. And now we can pass those genes onto other cells, creating the situation where they are now also resistant. If we prevent the drugs from ever coming into the cell, will the cell die? No. Or if we pump them out as quickly as they come in, will the cell die? No. So again, this is a bigger fight that has too many variables for us to really overcome. Even if we block the use of the drug, so if it disrupts things like a metabolic pathway and we switch different pathways in metabolism, that doesn't hurt us anymore. So the idea here is these drugs can be overcome by numerous ways. And so what still boils down to that question is whether the drugs are actually the correct answer for us. So this slide is really illustrating everything that we were just talking about. Enzymes that break beta-lactams literally break the use of penicillins. So another name for beta-lactamase is penicillinase because, again, it breaks the penicillin. The other thing is, again, preventing the drug from being taken up. If the drug isn't taken up, the drug doesn't work. If the drug is pumped out as quickly as it comes in, drug doesn't work. If we prevent the drug from doing its job, again, drug doesn't work. So let's illustrate this with some visuals now. So I've been talking to you about basically resistance being something that results from mutation. So there will typically be organisms in nature that are naturally resistant. But really the use of the drugs is what creates the favoritism of those resistant organisms. If I use the antibiotic on this plate, notice the red bacteria would be the only resistant bacteria. And as a result, after an hour, all the other bacteria die. But now we see the bigger problem. Because what if we let those ones that remain keep growing? Well, they increase in number and now become the dominant common feature. So with antibiotics, literally the more we use the antibiotics, the more we expose the microbes to the antibiotics, the less effective they become because more things become resistant to them. So with antibiotics, we're literally have a dead-end road. The more we use the drugs, the more quickly we're running towards the dead end of the road. So ultimately, the more drugs used, the more resistance. This is literally the mechanisms for survival of the fittest, right? So the more we apply this pressure, only the fittest or most resistant microbes survive. And so what should eventually happen through enough time and enough exposure, what will only remain in nature are the resistant organisms. So let's talk for a minute, because literally what I'm telling you is we're going to have to decrease our use of antibiotics. But whenever we stop and look at how we use antibiotics right now, we are completely and utterly misusing them. The vast majority of antimicrobial prescriptions are for infections that are most likely not even bacterial in nature. That instead of figuring out the exact cause, we're going to use a shotgun approach to try to cover our bases and make the person feel better. But the reality is viral infections are not at all affected by antibiotics. That taking antibiotics is actually hurting a person more than it's helping. So the reality is if we start using these drugs more appropriately, we can start conserving them for future use. Whenever we talk about the use of lots of serious amounts of antibiotics, it has to talk about the hospital. But here's a question I need to ask you. Where do all the very sick people go? They go to the hospital. So where do we administer the strongest, most potent drugs in existence? In the hospital. So what are we literally breeding every time we use these drugs in the hospital? We're breeding resistant microbes. And so where this usually starts is in the hospital. Again, Staph aureus resistant strains, so specifically things like MRSA, started in the hospital. Methicillin resistance started in the hospital. But over time, where do we find MRSA or methicillin resistance? It's not in the hospital anymore. It's even out in nature. The more we've used the drugs, the more we've favored the resistant organisms, and the more common they've become. But it doesn't even just have to be in humans. Really, the use of drugs, period, whether in a person, an animal, or just the environment. The more we expose microbes to the drugs, the less effective they become. And so I want to point an important stat out. The vast majority of antibiotics that are actually produced are not even given to people. They're given to animals. And do you think we're given these drugs before or after infection? It's before. So before the organism even gets an infection, we're going to treat it with antibiotics because that organism now can get bigger and fatter and make us more money and drive our food costs down. But really, at what cost to us in society? The more we use the drugs, the less effective they become, and the more problems we'll see in medical costs in the future. But this problem isn't even just here. This is a global problem. And what that means today is resistance may start in one country, but can easily be transported to another country through our global travel. So resistance isn't even something that we can just tackle in a local basis. We have to tackle it as a world. But that really means that we need to figure out the bigger problem. Where are all these most serious threats? Where are these big problems? But the bigger problem here is we don't know. We truly and literally have no idea how common resistance is because we don't have to even report them in this country, let alone in other countries where this can easily develop. The idea is we don't know how big this problem really is, but I can tell you it's much bigger than most people realize. That drug resistance is one of the most serious threats that we've really considered in the last 100 years, and it's literally our own creation. So do understand that if you need antibiotics, get antibiotics, but use them appropriately and with the most serious way you can. Follow the directions and do exactly what it tells you to do. Even if you feel better, even after a few pills, you need to take them all to kill as many bacteria as you possibly can to prevent the likelihood of resistance. But really, the answer is probably not what we've been doing in the past. The answer is probably something new. So finding new ways to target and destroy bacterial cells. Maybe designing drugs very specifically for one application that now allows us to have one single use so we don't push resistance across multiple species. But really, drugs are probably not the answer. Maybe figuring out ways to improve host immunity or to encourage the destruction of the bacteria through other means, maybe like viruses. Maybe even, again, the use of ancestral types of techniques. Maybe we need to move back to the use of more alcohols and acids to fight infection. But one of the things we can currently do that could be a great help is to encourage appropriate growth of a bacteria in the appropriate environment, to really push healthy biota. Because if good bacteria live where the bad bacteria would, there's no place for the bad bacteria. So having a good biota is actually a defensive mechanism against infection. The more we ensure people have the right bacteria in the right place, the less likely they are to get infected. Another important thing we need to consider is not just having the right bacteria in the right place, but purposely encouraging them to grow. And that's where prebiotics come in. Prebiotics aren't going to insert the bacteria in, but instead going to encourage the right bacteria to grow because we provide the right resources and environment for growth. So I've been kind of loosely illustrating this, but let's really just bring this to the point here. That really there are many times where a pathogen may be favored or promoted by the use of antibiotics. That there are some times where using an antibiotic creates more problems than they solve. The best example is C. diff. C. diff, which is Clostridium difficile, is something commonly found in many people. And what usually keeps C. diff levels low is competition from normal biota. So naturally, having good bacteria in the right place prevents C. diff from growing out of control. But what would happen if we used a broad spectrum antibiotic? What would it do to our normal biota? It'd destroy it. And guess who survives and now has all the space and resources to grow? That endospore forming Clostridium. And as a result, C. diff infections typically come on after the use of a broad spectrum antibiotic. But really the problem here was we lost competition. So what's a good way to reintroduce competition quickly and safely? Well, that involves the transfer of feces. Specifically, fecal transplant from someone with a good, healthy, normal biota that can be put in to somebody that no longer has the good bacteria to fight the C. diff. The next thing I want to really talk about is the fact that every drug has side effects, period. That there can be physical damage that results from using the drugs because the drugs are toxic. Another major concern is allergic reaction. Allergies exist. For every chemical or compound out there, somebody is going to be allergic to it. And this includes even drugs like antibiotics. The last thing, again, we just discussed was this disruption of the normal balance of normal biota. Naturally, again, this can create significant problems for the host or the person taking the drugs. So we illustrated that some drugs can actually be toxic. And I talked to you early on about this idea that many times antibiotics can affect things like liver and the kidneys, places where we detoxify or remove waste. But it also affects things like the gastrointestinal tract, the cardiovascular system, the nervous system, the respiratory tract, skin, bones, even teeth can be affected. So here's illustrations of exactly what I'm talking about. Even something natural like penicillin G, which is natural penicillin, causes skin abnormalities. And we actually consider this to be a very minor in terms of its side effects. We talked about tetracyclines causing basically the loss of good bacteria that can promote things like diarrhea and C. diff. We talked about things like polymyxins that can physically damage the kidneys. Or even other types of drugs like antifungals and antiprotozoans and antihelminths and antivirals. The idea is there's no drug out there that does not have side effects. every single drug has its risk, and you must weigh those risks against the reward. Tetracycline, as we illustrated, again disrupts things like the normal biota, promoting things like C. diff, but it actually also stains permanently the hard tissues of the body, which it doesn't sound bad for bones, but really whenever we talk about teeth, we're talking about a permanent gray to brown stain that stays with your teeth. This can also cause significant damage to the liver in a pregnant woman, or even cross the placenta and affect the offspring in terms of the deposition in the hard tissues. Diarrhea was another situation where we again lost good bacteria, allowing for the bad bacteria to take its place, including the C. diff example. I do want to point out this would often result in things like intestinal irritation or colitis. Colitis again being inflammation of the colon. Allergies were another threat that we were concerned with. Again, for every compound out there, someone's going to be allergic. And so using these drugs can actually promote allergic response in some people. And as a result, they shouldn't typically be given these drugs. Whenever we discuss C. diff, again, we talked about the loss of normal biota. But what results is what we call a superinfection. That the C. diff survives in the environment but is kept in low numbers by normal biota. If we kill off the normal biota, there's nothing to stop the C. diff from going out of control, and the result is a superinfection. So here's an illustration of exactly what you're talking about. Note the top image is going to have the normal setup, where we have C. diff, these red colonies, but usually what keeps the red colonies in low number is the normal of healthy bacteria, the white colonies. But what happens whenever we take the antibiotics? Well, we kill off the good, healthy white bacteria, leaving just the resistant red bacteria like C. diff behind. So what does that mean in terms of growth of C. diff? Well, it becomes incredibly common, and the result is colon inflammation that we call pseudomembranous colitis.